Good day. My name is Шагимолдина М О Н. I'm a biologist teacher in Nazarbayev Intellectual School of Astana. Today we'll look at a very interesting topic in biology, and it is the fundamental theory in biology. Exactly, our topic will be: how does cell structure relate to cell function, organism, organization. First, of course, we all should know our objects, what we are going to have at the end, what we should know at the end of this lesson. Describe the main ideas of the cell theory. Compare structure of animal and plant cells. Distinguish between prokaryotes and eukaryotes. If we will think about this thing, what is the smallest part of any living thing that still counts as being alive? What do you think? I think most of students who are already familiar with basic biology, you already will say that it is the cell. And if we will try to answer this question, is a leaf alive? How about a drop of blood? Blood is alive or non-living thing? Can we just keep dividing living things into smaller and smaller parts, or is there a point at which what's left is no longer alive? As you will see, there is much a limit. The smallest living unit of any organism is the cell. And of course, you all know from our lesson that we couldn't divide organism till the end. It has limit, and the limit, the unit of living things is the cell. The discovery of the cell, if we will look at the history, how scientists discover it, seeing is believing, an old saying goes, if you see a thing, you could believe it. In 1665, Englishman Robert Hooke used an early compound microscope to look at a non-living slice of cork, a plant material. And from that time started discovering the cell. Soon after Van Leeuwenhoek, observations by scientists made it clear that cells are the basic units of life. In 1838, German botanist Matthias Schleiden concluded that all plants are made of cells. The next year, German biologist Theodor Schwann stated that all animals are made of cells. And the next step in the history was in 1855, German physician Rudolf Virchow concluded that new cells can be produced only from the division of existing cells, confirming a suggestion made by German Lawrence Oken 50 years earlier. These discoveries are summarized in the cell theory, a fundamental concept of biology. You see here that before, in the cell history, lots of scientists worked on it, and for now we have the cell theory. What is the general rules of the cell theory? That all living, first one is all living things made of cells. It is very, it seems very simple, but in fact, it is very important. Why? Because before the discovery of the cell, of microscope, people didn't know that living things made of cells. For example, now we say living thing or living organism, the organisms which consist of cell. For example, we couldn't say the same about the virus, because viruses doesn't have the cell structure. Next rule of the cell theory is cells are the basic units of life. This is clear, yeah? All unicellular and multicellular organisms consist of cells. It is like little pieces of a big building or a big mechanism. Cells come only from existing cells. This is also very important for students to understand, because cells could come only from already existing cells, and at this time it will be very normal and very clever if you will have question like, where from first cell then? Yeah, if that cells, all cells come from already existing cell, where come from first cell? And it's like, first is the chicken or egg and chicken. In general, organisms are either unicellular or multicellular. Each cell is capable of carrying out biochemical reactions to support its metabolic processes. For example, grow, respond, and replicate. Okay, if we will look a little bit deeper into unicellular organisms, for example, in this picture you have very good examples: euglena, amoeba, and paramecium. If you look at this unicellular organism, yeah, it is a single cell that can carry out all those functions of life that are necessary to survive separately from other cells. They can rely on large surface area, volume ratio for exchange. The cell is specialized internally to obtain nutrition, carry out respiration, and produce. Yeah? Some biologists regard these organisms as acellular. Such biologists regard cells as interdependent units and not independent as these specialized unicellular organisms. Yes, if you look at unicellular organisms, unicellular organism has its own metabolism, and it doesn't link, or it could live itself, yeah, without another cells. But we couldn't say that about multicellular organisms. Multicellular organisms are large and have to specialize parts of their structure to complete the various functions that are characteristic of life. Cells within a multicellular organism specialize their function. Specialized cells have switched on particular genes expressed that correlate to these specialist functions. Yes, we said about multicellular organism, and now we are talking about first we said about unicellular organisms, and now we are talking about multicellular organism. For example, let's look at mammals. Yeah, mammals are multicellular organisms, and in mammals we have lots of cells, trillions of cells. And if you will look at one level higher, all cells in multicellular organism we could conclude into the different type of tissues, and different type of tissues could give us organs. Organs could give us organ system. Yeah, and why how come that cells in multicellular organisms different? Because it could specialize. How? This is very good question, because this one link with the genetics. As you know from genetics, if you don't know, this information will help you further link this information in genetics. Yeah. For example, if you look at one multicellular organism, you will know that in each cell of that multicellular organism, for example, trillions of cells, each cell has its own, has the same genetic material. But there could be different. For example, we could say easily that blood cell and muscle cell, it has a little bit different structure and different function. How come? Because some particular genes there are switched on and others could be switched off. Okay, these specific gene expressions produce particular shapes, functions and adaptations within a cell. Therefore, a muscle cell will express muscle genes, but not those genes which are for nerve cells. Cells that do not specialize are called stem cells, and now it is mostly the object of most contemporary research and debate. Yeah, and how do we call cells? I would like to emphasize this information. Cells in multicellular organisms which are not specialized, we call stem cells. We will look at stem cells a little bit deeper further with you. How we could organize, or the level of organization, yeah? Cells, tissues, organs, organ systems. Cells differentiation leads to higher levels of organization. In the cells of the tissue, some genes are expressed and others are suppressed, as we already said. Look at these levels of organization. Yeah, we will start from the tiny one, from small one to bigger. First, atoms. Atoms will give us molecules. Molecules will give us organelles. Now we'll look at those organelles. And organelles will give us cells. Cells, tissues, different type of tissues give us organs. Organs link together into the organ systems, and all organ systems give us the whole organism. Whole organisms in special area will give us populations, and population will give us communities. Then in this picture we don't have that, but then will be ecosystem and biosphere. You know what is the biosphere? Biosphere is the all living organisms on planet Earth. Okay, organelles. Do you remember from the previous picture there was level atoms, molecules, and then organelles. Yeah. Now look at organelles. Organelles are the discrete structures which the cell within the cell, yeah, inside of the cell. It has its own specific function. Look at this amazing picture of the cell. Now, if you look at this picture, in this picture you see anatomy of the animal cell, and here you could easily find out, yeah, at the end of this lesson you should be able to name all these organelles. Okay, this is animal cell. Next, this is that was the picture. This is schematic diagram of again, this is eukaryotic cell. Again, this is animal cell. Okay, we'll look at each of these organelles. Just look through. Label 1. In label 1, you have nucleolus. Label 2, it is nucleus. Label 3, it is ribosomes on the surface of the endoplasmic reticulum. Number 4, it is vesicle. Number 5, you see it is endoplasmic reticulum. Exactly, it is rough endoplasmic reticulum. Number 8, you see there endoplasmic reticulum, but it is smooth endoplasmic reticulum. Number 6, it is Golgi apparatus. Number 7 is the cytoskeleton. Number 9, it is mitochondria. 10, vacuole. 11, cytoplasm. And 12, lysosome. And 13, you see here centrioles. Prokaryotes and eukaryotes. How are prokaryotic and eukaryotic cells different? This is very good question, and because first you should know the difference. Yeah, cells falls into two broad categories depending on whether they contain a nucleus. The nucleus, in the previous picture it was labeled 2, plural nuclei, is a large membrane enclosed structure that contains genetic material in the form of DNA and controls many of the cell's activities. Okay, because of this very part of cell, yeah, because of the nucleus, we could divide all living organisms into two, prokaryotes and eukaryotes. Prokaryotes, we say them bacteria, and eukaryotic organisms are protists, are fungi, plants, and animals. Prokaryotic cell. Yeah, now we'll look at the general structure, the basic structure. Significant structural differences exist between prokaryotes and eukaryotes. For example, if you look at this picture, the main distinguished features between prokaryote and eukaryote, as we already said, is the lack of a true nucleus in the former. Okay, if I'm so quick, I will a little bit make my speech slower. Look at this picture here. Here you has bacterium, yeah, which live in our digestive tract, and this bacteria, most of scientists, they crazy with it because they like use this bacteria in their laboratories. E. coli, we call it E. coli. E. coli first has outside, it has capsule, yeah, do you see that? Dark gray, yeah, capsule. Why bacteria need capsule. Most of bacteria has capsules, yeah, have capsules. Capsule protect them from the outside environment, bad conditions. Then it has cell wall, and then plasma membrane. Then inside you could find cytoplasm, yeah, where you could find cytoplasm. And then you see here nucleoid, DNA, plasmid, yeah, circled one, mesosome, it is enfolding inside, yeah, mesosome, and ribosome. The ribosome exactly is 70S ribosome. And you could see here flagellum. Some bacteria have flagellum. What is the function of flagellum? It helps. It helps bacteria to move. Okay, not all these features are present in all prokaryotes. Not always. Okay, and if we will, we already named all of this. Now let's look at our a little bit come back. Yeah, structures of prokaryotic cell. This topic itself is also very big topic. Yeah, that's why for now important to remember the basic parts of prokaryotic cell. Okay, do you remember all the parts of prokaryotic cell? Okay, if yes, it's very good. Go further. Now we'll look at eukaryotic cells. Here in this picture you have liver cell. Eukaryotic cells, structure and function. I will name the basic ones: nucleus, mitochondria, plasma membrane, cytoplasm, Golgi apparatus, chloroplast, vacuole, and cell wall. First, we'll look at the nucleus. Yeah, do you remember it was labeled 2 in the first animal cell picture? Nucleus has a double membrane with pores. It has double membrane, 2 membranes, yeah, and nucleus controls the cell via expression of genes. This organelle, this part of the cell, this part of the cell, like a boris inside of the cell, I could compare it with the boris because it has genes, and those genes give information to synthesize proteins, exact type of proteins. That is why it is very important. Here in the you have 2 pictures. First one is the picture, yeah, from the microscope, electron microscope, and the second one the schematic version of that. DM means double membrane, and P means nuclear pore, PM means plasma membrane, nucleolus, you could see that inside of the nucleus, and cyto means cytoplasm. Plasma membrane. Plasma membrane controls what enters and leaves the cell, and the function is protection and integrity. If you look at the plasma membrane divide cell from the outer environment, or in multicellular organisms it divide exact one cell from other cells, and it control what will enter in and go out, leave the cell. Yeah, the function of plasma membrane is also very, very important. If you will look at the structure of that, you see it is scientists call this model fluid mosaic model, because it is fluid. Yeah, it's clear, I think. And mosaic because it has structure which connect different tiny parts together. Okay, if you look at this picture carefully, you easily could see that it consists of two layers. Yeah, two layers in red in this picture. We call it phospholipid bilayer. Bi means two. Each of these phospholipid bilayer consists of phospholipid molecule. If you will look and try to find information a little bit more, you know that it has phosphor and lipid, yeah, molecule. And this exact structure is very important because it has hydrophobic and hydrophilic sides. It is very important. In this picture you could find it. For example, hydrophilic heads. The phosphor heads, it is hydrophilic, and the tails is hydrophobic, don't like water. And this one, this structure helps cell to fix its shape, and it prevents cell to mix in with other cells. Yeah. Okay, the next one you see here, protein channels in blue, transport protein. Yeah. And you could find here peripheral and integral protein. Integral protein embedded inside of the cell. You could see that it goes through the cell, membrane. Sorry, it stays in both sides. Yeah, in the bilayer. And peripheral protein, it not embedded fully. It is in one layer only. What is the function of this protein? Integral protein mostly has function of channel. Yeah, it will control what will in and out by active transport mostly. But sometimes, of course, passive transport also. Then you could see glycolipids here, glycoproteins, and then you could find here cholesterol. What is the function of cholesterol? It will keep the shape, yeah, of the cell membrane and all the cell by it. And also you could see those long filaments of cytoskeleton, which also the function is making it stronger. Yeah. Okay. In the top you could, it is the extracellular fluid, and the second side is the cytoplasm inside of the cell. Okay, next organelle is the vacuole. The vacuole is a storage area in plants for amino acids and sugars. Vacuole we could find in both in animal cell and in plant cell, but the difference between plant cell and animal cell vacuole is that in plants, mostly in old plant cell, in adult plant cell, let's say like that, the vacuole usually it is central and big, but in animal cell it mostly small, and it could be some, yeah, not one big vacuole. Okay, and what is the function of vacuole? Vacuole could store water, some another organic materials, and there also could be pigments, yeah, which will give, for example, in beet, it give red color. The tonoplast is a membrane like the plasma membrane. It controls what enters and leaves the vacuole. Tonoplast is the membrane of vacuole, and that's all. Okay. Ribosomes. Ribosomes, proteins are assembled on ribosomes. Ribosomes are small particles of RNA and protein found throughout the cytoplasm in all cells. This ribosome we could find in both in prokaryotes and in eukaryotes. Ribosome usually has two subunits, small and large subunit. Now, in protein synthesis, in this topic, you will look at the structure of ribosome very, very deep. Okay, and in prokaryote cell, you could find ribosomes 70S, 70 Svedberg, and in eukaryotes it is 80S size. Rough endoplasmic reticulum. We said that endoplasmic reticulum we have two types, yeah? Rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum consists on its surface lots of ribosomes, and it is a complex interconnected network of membrane tubes. The surface is covered in ribosomes where proteins are made for secretion, and attached ribosomes make proteins for internal use. Yeah. For example, the protein which make this on the surface of this rough endoplasmic reticulum mostly are for uses of externally, yeah, but of course we could find ribosomes which are free on the cytoplasm. Those ones make protein for its own cell, not for neighbor cells. Okay. Golgi apparatus. The Golgi apparatus modifies, sorts, and packages proteins and other materials from the endoplasmic reticulum for storage in the cell or release outside the cell. Golgi apparatus, yeah, it is now named according to the scientist Golgi, and it consists of tubes, vesicles, and spaces between tubes. and it allow to transport protein. Yeah, the function is modifying and sorting and packaging, yeah, of proteins. Making proteins. Look at this picture, please, carefully. You first are in green one, labeled one. Yeah, it is nucleus. Nucleus will give information to ribosomes with help of mRNA, will give information to synthesize protein. If you look at the rough endoplasmic reticulum, which gives number 3, and exactly number 5 represents ribosome on its surface, on this surface, this ribosome will synthesize protein. Then, with the help of vesicles of Golgi apparatus number 7, this vesicle will give that protein into the Golgi apparatus. Golgi apparatus will modify that protein, and then with the help of vesicle it will go number 12, you see, then you see number 14. Number 14 represents that protein which was modified, go in extra cellular, outside of the cell, and it will be used externally. Yeah. Okay, this is very short about the making protein synthesis, but at least you will have an idea of that. Okay, now chloroplast. In plants only, only this is the site of photosynthesis. Light is trapped by chlorophyll found on the thylakoids. Carbohydrate is made using the trapped energy and carbon dioxide in the stroma. Oxygen is produced as a waste gas from the process. Chloroplast, here you have two pictures of chloroplast. Yeah. The second one is the diagram of chloroplast. First one is from electron microscope. Chloroplast also double membraned. It has two membranes. Inside it has stroma, the liquid-like stuff. Then you could see here DNA. Then you could find here thylakoids, oil droplets, and starch grains. Exactly, light, yeah, the first energy will trap by the thylakoid, and then it will be converted into the chemical energy. Remember please that chloroplast consists its own DNA, because this is important. After I will explain why after mitochondria. Mitochondria is the site of aerobic respiration. The matrix is the site of the Krebs cycle. Oxidative phosphorylation occurs on the crystal membrane. Very active cells usually have a lot of mitochondria. For example, our muscle cells. If you look at the structure of mitochondria, you see double membrane there. Then inside of it you see matrix. Then you see infolding of membrane, which is crystal. Exactly, reaction happen in crystal. Why do we need crystal? Why the second membrane of mitochondria is enfolding, and why it is important? Because it will increase the surface area for respiration. That is why. And of course, in this picture you couldn't see, but inside of mitochondria you could find DNA. Do you remember which another organelle has DNA? It was chloroplast. Chloroplast and mitochondria have its own DNA. This information very interesting because these two organelles synthesize its own protein, and it could control its own reactions, yeah. And some scientists think that maybe chloroplast and mitochondria is the symbiosis between the cell and these organelles. They guess that maybe chloroplast and mitochondria was independent organisms at the beginning, and then in some stage of their life they enter into the cell and then stay there. This is very interesting, and there are some facts which say that it's true, or it is only some guessing of some scientists. And one of the most interesting aspects of mitochondria is the way in which they are inherited. In humans, all or nearly all of our mitochondria come from the cytoplasm of the ovum or egg cell. This means that when your relatives are discussing which side of the family should take credit for your best characteristics, you can tell them that you got your mitochondria from your mom, because it's true. Cell wall. The cell wall supports plant tissue composed of a fully permeable wall of the cellulose, important structure in establishing turgidity. Cell wall we could find in plant cells and in bacteria. Now let's compare prokaryotic and eukaryotic cells. Prokaryotic cell smaller and eukaryotic cell larger. Prokaryotic cell always unicellular. Eukaryotic cell mostly or often multicellular. No nucleus instead has a naked loop called a nucleoid. Eukaryotic cell has true nucleus with a linear DNA, which we call chromosomes. Yeah, DNA with the histone. No membrane-bound organelles. In eukaryote we could find membrane-bound organelles. In prokaryote cells, ribosomes are 70S, and in eukaryotic it is 80S. Prokaryotic cell doesn't have mitochondria, instead has a mesosome. But in eukaryotic cell we could find mitochondria. Prokaryotic cell divide by binary fission. Eukaryotic cell divide by mitosis or meiosis. Prokaryotic reproduction mainly asexually, however, there are some non-meiotic forms of gene exchange, such as conjugation, yeah, and eukaryotic cell reproduction could be asexual or sexual. Prokaryotic cell has many metabolic pathways: fermentation, nitrogen fixation, and photosynthesis. Eukaryotic cell, common form of respiration involving organic molecules like sugars. This is the basic difference, yeah, between prokaryotic and eukaryotic cells. And here I will give you the summary of this whole lesson. Here you could find first structure, function, and could we find it in prokaryote and in eukaryote cells, yeah, and all the organelles you could find here. Okay, now let's look at homework. You should create a Venn diagram that illustrates the similarities and differences between prokaryotes and eukaryotes. Compare light and electron microscopes, yeah, the advantage and disadvantage of both. And the cell as a living factory, you should make analogy between the factory and the cell. Here you could see bibliography. All information comes from these resources. Thank you for your attention. I hope that you enjoy our lesson. See you next lesson. Bye.